Welcome everybody to SynBioBeta Live. This is the first of what we hope will be many live shows from California where we delve a little bit deeper into what's going on in the synthetic biology industry. I'm joined today by Tim Gardner, the founder and CEO of Riffin, and Laura Antipov Jennings-Antipov, who is the Director of Science at Riffin. And we're going to be doing a demo of Riffin later on so you can understand how you can go from machine learning or from lab data to machine learning in 30 seconds. We've also got some great guests today that we've filmed ahead of time. One of them is John Conway, formerly of Accenture, and he's going to be talking about how every company needs a big data strategy. We've also gone down to Hayward, California to visit Aaron Hammock, who's the CEO of Epibiome, and Epibiome's making new antibiotic resistance drugs using phage, and they're also using Riffin, and we're going to look under the hood and see what they're actually doing down there. So I want to make this as interactive as possible. We have a live studio audience at the background here, Kevin and Garrett. And Kevin and Garrett are going to be on the Twitter and on the YouTube comments, and also Mariana, who's on our team, is going to be on the email. So you may email us at live@synbiobeta.com. You may add some questions to the comments feed on YouTube, and you can tweet us at SynBioBeta. And there will be a free ticket to SynBioBeta in October in San Francisco for the best question of this episode. So do send us your comments, do send us your questions, and if at any point during the live broadcast you would like to sign up for a demo of Riffin, then you can also email hello@riffin.com, and they'll set you up with an account. So I wanted to start with a few news headlines from the digest that went out this morning. We saw that Ginkgo Bioworks made it into CNBC's top 50 companies, most innovative companies, which was great. Also, LanzaTech was in that list. We also saw that Pivot Bio is expanding their leadership team. Pivot's the company that's engineering the microbiome to make new microbes that can fix nitrogen for crops. Biosyntia, the Danish company that's engineering yeast to make vitamins, has just closed a €4 million Series A. And Indie Labs, a company that's making some really cool hardware for gene therapy, has also just closed a 2.6 million seed series. They're an indie bio company, and I believe that all of those companies are going to be speaking at SynBioBeta in October. And one of the other pieces of content that we put out this week was a really in-depth interview, a really nice in-depth interview with Bill Peck, one of the founders of Twist Bioscience and the CTO of Twist Bioscience. And that was all about why the future needs to be written in DNA. So that was the news that came this week. In particular, I wanted to introduce Tim Gardner. As I said, Tim is the founder and CEO of Riffin, and you may recognize Tim from such famous papers as a bistable toggle switch, published in Nature in 2000. Correct, Tim?
Genetic toggle switch.
A genetic toggle switch. Yeah, that would be quite important, right?
Yeah.
But in particular, I want to thank Tim for and do a little pop quiz. Can you tell me what this is?
Oh, this is tough. It's a guinea pig.
It is a guinea pig. Yeah. So this is what Riffin is today, and what Tim and Laura have been, because this is the very first live show that we're doing, and anything could, and I hope a few things do go wrong in this live show. Clearly, we could in this day and age have recorded this, but I wanted the excitement and the thrill of doing something live, and that's why we're doing it. So thank you. When I told, we've been riffing on the format of this, if you excuse the pun, for quite some time, and I told him that it was going to be a mix between Wayne's World and The Daily Show, and so that's what we're promising you. So if you haven't seen Wayne's World, from a movie from the 80s, then I suggest you go see it. And Tim's question was, well, do I get to hold the drumsticks or the guitar? So
neither was the answer.
Neither was the answer. Yeah. So anyway, but I do appreciate you being the guinea pig in this, and. In particular, so let's just dive in a little bit. We're here to talk about Riffin, lab data to machine learning in 30 seconds. Can you tell me a little bit about why you started Riffin and where the idea came from originally?
Yeah, so Riffin is really the synthesis of about 20 years of hands-on work in biotechnology R&D. So I started out in microbial genetics and metabolic engineering. I spent some time as a professor focused on machine learning and statistical learning applied to microarray data, and then I eventually ended up at Amyris, where I was leading R&D and developing genetically engineered yeast and bioprocesses and scaling that to 200,000 liter manufacturing. And the one thing that struck me throughout all of those experiences was the general problematic nature of how data is handled. It's fragmented, it's siloed, and it's often of very low quality because people have not understood the sources of error in their experiments. As a result, people make really, frankly, bad decisions, not because they're not smart, but because they're not using data as effectively as they could. And after those 20 years, I decided it was time to do something about it, or at least make an attempt, and that's really what Riffin is. It's an answer to the problem of fragmented, siloed, low-quality data in R&D organizations.
Excellent. So we're going to see some of those examples of some of the companies that are using it and what they're using it for. We talked previously about what it is that scientists spend most of their time doing, and I think you did a study on this. Is that right? You have some
we
data?
didn't. We found a study on it.
Okay. And what did the study say?
Crowdflower published a study where they surveyed data scientists who, on average, spent about 80% of their time cleaning up the data, not analyzing it. And that's a data point that just reflects a common knowledge and an intuition I think most people have, is how much time they just spend doing the housekeeping, but not actually discovering new things from the data.
Got it. Okay. Great. Laura, in some of your time speaking to some of your customers, how have you seen them change over time in terms of how they spend their time and what they were doing in the past and what they're now doing?
Yeah, so I've seen, I mean, even in my career, I've seen this where the scientists are just really hunting for data. And they're spending so much time just trying to look through their spreadsheets to try to understand, what does this column mean? I labeled it like this. I don't know what this means now. And they're going back trying to, retrospectively pull it all together. And then I've seen them using Riffin be able to just say, okay, I'm ready to do the analysis. I just click a button and it's all joined in there. There's just eliminating so much wasted time, trying to track down the data using Riffin.
Got it. So we're going to see in the demo exactly how we can do this and how you can save time. But it's not just us saying it. We went out and we spoke with John Conway at his farmhouse in Pennsylvania. John is the former Global Director of R&D Innovation and Thought Leadership at Accenture, and he's soon to be the head of R&D Information Technology at MedImmune. In fact, I think he started on Monday. He spoke to us last week about some of the activities that are going on, or some of the cost savings that can be made if we can make this process more efficient. So let's cut to John Conway's clip and let's see what he had to say.
Today, the inherent cost of doing research and development is staggering, and there's a lot of waste in research and development. By driving this forward, the efficiency gains alone could start to dramatically reduce this cost and allow us to even focus more on new discoveries, new biology, things we don't know today. We don't know a lot when it comes to biology and chemistry, and there's a lot to learn. And I think this is where some of these efficiency gains will allow us to even drive this further.
So what we heard John say was how innovation is being sped up by this ability to gather the data and to push the data into machine learning tools. How have you seen that actually happening in reality in some of the customers that you've worked with? What are some of the big things that we've seen from their implementation of Riffin? Maybe you could give just a couple of examples, Tim.
Yeah, one of the amazing observations we've seen is that every time we implement Riffin, usually within the first experimental data set, we found unexpected relationships or errors or deviations that they'd never seen before. In one case, we saw a situation where preclinical studies were being conducted at two different sites with the same compound, the same protocol, and the answers they received were completely different. So what Riffin has really immediately helps to do is uncover those anomalies and defects in data sets that actually lead to artifacts in decision making and a lot of wasted effort downstream, some of the costs and pain that John Conway refers to. So we start by cleaning up that, and once the data set has been cleaned up and the experimental processes are humming along more smoothly, we can start to uncover the physiological and causal relationships between the underlying biological or system variables.
Got it. Okay. Well, great. Let's. So we looked at R&D becoming more efficient, and can we get the audio on those John Conway videos? So let's now look at one of the other industries that John has experience at. He was talking about the efficiencies that were made in chemistry and how that applies to biology. So let's see what John had to say there.
If you think about the impact with chemistry, for example, it takes about 10 years for a chemist to become proficient in their craft. What if all this knowledge, machine learning, has provided this environment that a chemist now could do that in 5 years? So now we're at a 50% efficiency gain. That's ginormous. How do we keep taking that in biology to further new discoveries and apply those learnings in biology? Because we don't know a lot in biology today. That is where the future is going.
I was referring to the advances that we made in chemistry and how we can speed up the efficiency in those industries. If we look at how biology is engineered right now, if we go back, let's say, to an academic lab in grad school or even a startup company, how do you think that's changing over time in terms of how people think about their experimental design? Or any comments in how maybe synthetic chemistry techniques and tools could be applied to synthetic biology?
Well, one of the transformations in chemistry that I'm aware of from 15 or so years ago was an increase to massive scale. So combinatorial chemistry data sets exploded to tens of thousands, to hundreds of thousands, to millions of compounds in very high throughput screening. When you're working at that scale, it's extremely easy to end up with false positives and false detections. So first of all, you have to get your assay systems running with incredible precision. But the other thing that does is it gives you the opportunity to assess a larger number of variables in combination. And that brings you to trying to understand data sets, multi-factor, multivariate data sets that frankly you can't interpret by looking at a single bar chart. You need machines to help you see the combined impact of all those variables. So that leads to the need for better experimental design, better data collection, better quality, and machine learning. And those are all driving factors for the architecture for Riffin and why we built it the way we did.
Got it. So John Conway at Accenture was talking about model quality data and why that's really important for R&D. So I asked John, what is model quality data and why is it important for machine learning? Let's see what he had to say.
Yeah, model quality data is data that is produced in an environment that has more constraints around it. The metadata is captured, the methodologies, the versions of software, the attention to detail when it comes to the metadata and contextualization of the data, and then making sure that any variances in that data are captured. A lot of times if you don't do that and you start to aggregate data, you realize that, that these different data sets aren't working, and I can't get the results I want out of this particular algorithm or whatever that I was looking for. So it's really about the rules and the constraints that you put around your data and how you produce it, because it's really about if you have consistent rules, you can consistently update or change that data or use that data. And that's a really critical thing when it comes to machine learning, because you're getting at the level of noise in your data sets, and if your noise levels are much larger than they should be, you're not going to be able to apply these tools and get the results you want, and that's going to be a very costly mistake for a lot of organizations.
Laura, maybe you can give us some examples of some of the organizations that you've helped implement this and some of the impact that it's had on them.
Yeah, so I've in particular recently been working with, and I got to see, the inefficiencies with which they were collecting their data and really how Riffin could help them. So, I've seen them go from collecting data, literally on non-mindable PDFs, to pushing that data into structured data sets on the cloud, accessible to team members around the world, and then we can really see how quickly they can pull trends out of their data and really increase the efficiency of their processes.
Got it. Okay. Tim, any thoughts?
I just want to reinforce what John Conway said and how much that resonates, that it's critical to have good quality data from the beginning, because it's basically the old adage, garbage in, garbage out. And when you feed garbage to a machine, you really just magnify the scale of the problem. To put it in other terms, your signal, the teeny physiological signals that you're looking for that might be the discovery of the next drug, if there's too much noise, it's buried and you'll never see those. And the most insidious problem in R&D that we're mostly unaware of is not so much the false positives, the artifacts of your experiments. It's the missed opportunities, the false negatives, the missed discoveries that just go right under the radar and nobody has any idea. And that's money out the door.
Is it just bio companies that you're working with, or are there other R&D organizations who are implementing Riffin?
No, we work across the board in process-based R&D, so chemistry, material science, industrial biotech, pharmaceutical, anything that is recipe-driven, laboratory type of work. The boundaries of where Riffin plays, we don't currently operate manufacturing lines, so it's not manufacturing software. We don't do clinical trials. And in the very, very, very early stages of discovery, when it's almost completely freeform, that's a little earlier for our software. It's after maybe the kicking things around that you start to get some rhythm to your R&D that we apply.
Got it. And then once all the data's collected, then the machine learning takes over, and that just solves all of our problems, right?
That's right. I mean, it's a magic bullet. If you just take machine learning and just apply it to anything, it's magic. It's like Occupy Oakland, Occupy. No.
So this is one of the questions I asked John Conway, that AI and ML, machine learning, are going to solve all of our problems. This is what he had to say.
No, machine learning and AI are not going to solve all of our problems. They are part of the solution, and this is where the hype, where we're at right now in the hype, is that they are a tool to be used. And what is going to solve a lot of problems is that people adopt a scientific data strategy and they adhere to it and they sacrificed and they commit to it. And tools like AI and ML are going to be there to consume that higher quality data and now produce even more efficiency and quality coming out of that. And that's where you're going to see the big benefits.
So ML isn't going to solve all your problems, but certainly getting your data into this format where you can use machine learning is going to be a big boon. So, so Laura, I wanted to pass over to you because we did a demo that we pre-filmed that we're going to show folks, and we're really going to get under the hood and show them how Riffin can be used. So let's run the demo, and then we'll come back and we've got a couple of questions that we can go through. So this is Laura and I doing a demo earlier. Okay, so we're going to do a demo now, and the demo that we've chosen to show you is Succinate Production. So Succinate, you might have heard of. It's a pretty important metabolite in central metabolism, and it's also being made by a couple of companies in the synthetic biology industry. One of them is Lygos, the other one is Myriad, and it's used as a precursor chemical for a whole number of things, including things like paint thinners. So it's a pretty important chemical that currently is being made by a petrochemical industry, and the synthetic biology industry is picked this up and is converting sugar into succinate. So talk us through a workflow that a company might use for the production of succinate. And maybe you can just pull up Riffin and just show us how you start off. Maybe grab something and throw it into this framework that you're seeing. So if I wanted to start from scratch and just pull something up and throw in something, how would I do that?
Yeah, so here's a process that's partially made.
Okay.
Right. So we've got inoculate media, pellet and wash cells, and then batch fermentation. So if we want to say, okay, what do I do after this? Well, maybe I'll take some samples. So I can just add a step here and say, okay, this is my take sample step. Oops, if I could spell. Here we go. And then we can say, okay, what do we do after sampling? Well, maybe we're going to do metabolite analysis.
Got it. So you can build up this visual pathway of your experiment. And so go back to the one that you made earlier. So that's the whole thing that you've got here. So you've got inoculate media. So you've got engineered strains that you've made. They all produce succinate. You want to find out how can we maximize the yield, and what are the variables that affect that yield production?
Yeah, that's right. So what we're going to do in Riffin is transform their protocol into a visual platform for experimental design.
Okay.
So you'll see here that here is their procedure represented as all these different steps.
Okay.
And so, you see it visually. There's also a platform for recording videos. So here's a video of doing, sterile inoculation.
And that might be if you've got a difficult protocol and somebody new joins the lab and you don't want to have to show them every time, so you can just take a video on your iPhone and upload it to Riffin.
That's right. We've gotten feedback that this is a very useful tool, especially as there's turnover.
Right.
You don't have to keep retraining people. You can just say, oh, just go check out the Riffin process. There's videos there.
Got it. So in this example, again, we've got different strains and of yeast in this example. We're pelleting them, we're washing them, we're putting them inside a fermenter, inside a bioreactor, and then we're going to take samples over time. And then we're going to look at the output of these experiments. And so you've got three different outputs there. So just talk us through what those are.
Yeah, so this is measuring the ODs, that's the optical density. So that's for you to measure the growth rate.
Okay.
This is HPLC analysis, where you're going to look at the metabolite profile.
Okay.
And this is the biomass dry weight analysis. So that's where you're putting the cells on a filter and drying it, and then you're looking at biomass production.
Got it. So HPLC, high performance liquid chromatography. So you're actually looking at the succinate and how much of it is produced.
Yes, that's right.
Molecular analysis.
That's right.
Okay, great. So now show us an example of the old way of doing this. You had some data that you showed me before. If you can pull that up. Okay, so I'm very familiar with this. This looks like just my PhD thesis and what I would do in graduate school. So you've got 4 strains there, and you've got a few different conditions that you're growing these strains under. And you can't quite see, or you can't see on this monitor, but the strain 1 is orangey and strain 4 is red. It's a little bit of the same color. So you've got and then the circles are the media, the different media, and the triangles are the different pH. So you're trying to find out, your goal, if you so choose to accept it, is to find out which of these strains performs better under which condition. So, and this is what I do in graduate school. I kind of put my finger into the air and I pick one and think that maybe it's a little bit more than the other, and I choose that, or I choose a parameter to then optimize or something else to go after. That's the old way of doing things.
Yep, that's right. Yeah, so here, this is typical of how many of our customers are analyzing their data today. So they just plot it, and then they colored by strain, and they said, okay, I varied strain and I varied pH, and I want to understand the contribution of those two. And so what they see here is that the circles and the triangles are on top of each other, indicating that no effect of pH. But there appears to be an effect of strain, right? And the orange, which you're right doesn't look so hot here, it looks orange on my screen, is that looks like the best strain, so that's strain 4.
Okay.
But what I'm going to show you today in my case study is that strain 4 is actually not the best strain, it's actually strain 2.
Okay.
And you would never know this just by plotting the data, as many of our customers are doing.
Got it. So go back to now the big plot. So you've now run this, you've entered all of your data and all of your parameters into this workflow, and then you've got the data, the data that comes out. So now, can you show me the machine learning? Where does that come into play?
Yeah. Okay. So what we're going to do at Riffin is automatically annotate and automatically join your data.
Okay.
The moment that you collect it. So let me show you how we do that.
Okay. Great.
And so it all starts with the process flow diagram, as we've already walked through, and then the next step here is to define your inputs and outputs for those steps. So here we've got cell bank and media as the input to inoculate media, which creates a seed culture. The seed culture gets transformed, or it gets passed through after washing into the batch fermentation step, which creates a cell culture that then gets sampled and sent to all these different analyses. So that's the material and sample flow across the steps. And then the third component here are the properties.
Okay.
So if we look down here, you'll see that, for instance, cell bank, which is an input to inoculate media, has a number of properties like lot number, volume, strain name, and these are placeholders for your data.
Okay.
So what you're putting together here is a reusable template for recording experimental data. So you're saying that when you record data on experiments, you want to record lot number, volume, name, etc. And so what you've done here is really created a data collection engine, because what looks to you and I like a process flow diagram that I might draw on Visio is actually telling Riffin on the back end to join all the data. So when the seed culture, which is the output of step 1, becomes the input to step 2, Riffin knows to join that data. And then when this output becomes the input here, we join the data. So let me show you what that looks like in an actual example. So as I've stated, this is the template for data analysis, and here, template for data collection. Here's an actual experiment where we recorded data on an experiment. So just to orient you, you saw before that those properties were going down the screen, and here in experiment mode, they're going across the screen. So, so here we've got 2 strains that were being used to inoculate media, just to show you the flow of data here. So that one strain is now being split and to inoculate 2 bioreactors at 2 different pHs. And then if I just click on between batch fermentation and HPLC analysis, you'll see that strain 1 is now being flowed into 15 samples were taken for strain 1, and then you'll see 15 samples for strain 2, etc. And so this, to me, is a really good interface for visualizing how the data are connected. But let me show you what it looked like before using Riffin. So, so if we come here, I'll show you how our users are typically recording their data. So this is a batch record, and so this contains a lot of procedural information about how they're running their experiment. But what you'll notice here is that there's a lot of data here. So so like, you know, the lot numbers of the strains, the lot numbers of the media, there's pHs, shaker speeds, etc. All of these are variables that could affect my key outcome. So my key outcome here, remember, is succinate yield. Any of these could affect the yield, but the way that they're being recorded now is a non-structured, non-mineable format. So if later on I want to try to correlate, say, lot number of my strain with the yield of my succinate, there's really no easy way to do it.
So go back to Riffin and just show me when then the data comes out of it. Show me in particular how that gets converted into something that you can intuitively use then.
Yeah, exactly. So once I have all of my data in, all I need to do is just come down here and say export data from this experiment.
Okay.
Or if I want to join all the data across all the experiments on the process, which is most likely what you want to do because you want to look at the data in aggregate, you can just come here and say export data from all of the experiments.
Got it.
So I'm just going to do that right now. So I'm going to download this Riffin generated file. And I'm going to open this. I'm going to open it in JMP. So Riffin generates a CSV file that you can open with any program. So I'm opening it with JMP. You could open it with Spotfire, with R, with Python. I downloaded it directly from our user interface, but you can also pull it out programmatically via the API.
So what Riffin's allowed you to do is to really look at everything visually, collect all the data, tag it, annotate it, collect all the instrument data, all the strain data, all the metadata associated with it in one place, and you wouldn't have been able to do that before.
Yes, that's right. So after you put the data in Riffin, it'll get automatically joined. Remember, let me just take a step back. Remember via these connections between the steps.
Yeah.
So if I go here, when I put in my batch fermentation data, and when I put in my HPLC metabolite data, it's going to get automatically joined to all the relevant data upstream and downstream.
Got
so
it.
that the end product looks like this.
Got it. So now we're back into JMP, and now we can do the analysis. So yeah.
So just to orient you here, so this data table has, you'll recognize these all the steps of my process.
Got it.
And then if I expand this, you'll see that all of those properties that you saw in the process are now in the data table.
Got it.
So all that, all the metadata, all the procedural data I talked about earlier, the lot numbers, you know, the pH, the temperature, the fermentation data, the HPLC data, all here.
So show me now which ones correlate and which ones don't. How do I find that?
Yeah, so let's do that. So we'll do our correlation analysis. So this is back to that same plot, right, that we talked about before.
This is the old way of doing
This is the old way of doing things.
Okay.
But when you have a Riffin-generated data table, it allows you to do these correlation analyses.
Got it. So I'm seeing your succinate yield there versus the theoretical yield. Okay, that's showing that the parameters are aligned. Mm-hmm. What have we got over there?
Yeah, so what this plot is telling us is that the variables in our model are accurately predicting the succinate yield, and the variables in our model are strain, pH, and temperature.
Got it. So we can see a very strong correlation there. no correlation there. So this one is
This is the strain. So there's an effective strain
versus yield. This is pH versus yield, so no correlation with pH.
Right.
And then this one is temperature, so strong correlation with temperature.
Right.
Okay.
And so if you noticed before when we were just plotting, the only two variables that they purposely varied were strain and pH. And so what looked like before an effective strain was actually confounded by the fact that it was an effective strain and temperature. So temperature they didn't mean to vary, but maybe operator error or drift in their instrumentation, they actually performed these fermentations across different temperatures.
Got it.
And so now they're seeing that actually there's a large effect of temperature. So when you correct for that effect of temperature, this is the actual order of the strains in terms of their succinate yield.
Got
So
it.
strain 2 turns out to be the best. If we go back to this plot, strain 2 is in green here. It looks like it's worse than 4, but that was only because these were run at different temperatures. So when
Got
I
it.
take that into account, now I can accurately identify the top producing strain and move that into scale up versus the mediocre one that I would have chosen before.
Got it. So if you can just go back to the Riffin screen again. So again, what you're seeing is a visual workflow, and you can see here all the different steps of the protocol here. You can see the data is annotated, the metadata is attached to it, and then the data that it spits out the other end, you can then run these statistical machine learning algorithms on it. And there's basically hidden, the secret, the answer of which strain to use was hidden inside this data. A human brain couldn't analyze it. You can't see it by a visual analysis. The machine learning can, and it pulls out and tells you which parameters to optimize and how to increase your yield, and it tells you which experiment to do next.
Yeah, that's right. So one of our customers prior to using Riffin, they said, we work on what we know, not necessarily what matters. And with Riffin, we allow you to flip that around, and we allow you to work on what matters, not necessarily what you knew going into the experiment. Because once you do the analysis, you didn't know that temperature had such a large effect. Now we know it does. So now, moving to scale up, we know, hey, we need to control for temperature. And in fact, this looks like a good variable for us to vary to try and increase the yield of our strains.
Got it. Excellent. Laura, thank you so much.
So if you would like to take a demo, take a test drive, then just email hello@riffin.com. And if you have any questions for Tim, Laura, or myself, then please drop us an email at live@synbiobeta.com. You can also get in touch with us on Twitter. And also you should be able to leave some comments in the YouTube feed, although somebody's told me that that's not quite working very well. So Twitter is probably the best way to get a hold of us, and we'll be asking the questions live here. There's a free ticket to SynBioBeta in October for the best question. And we did actually have a question come in from Eric Merle. And Eric said, what size of organizations are best for implementing machine learning, and also what size of organizations are best for Riffin? Tim, any thoughts?
So we've designed Riffin to scale from a single individual user up to thousands of users. So the answer is what size? 1. And the whole point of designing Riffin the way we did it was to democratize machine learning and data science, to put it in the hands of the scientists who are generating the data rather than offloading it to somebody else who doesn't necessarily have all the insight and intuition. So if you're excited about machine learning and you want to get your hands into it, Riffin's a great way to do that. You do need some knowledge, and Riffin also provides training for individuals who want to understand the principles and practices beyond just clicking the buttons.
Got it. Great. So Laura, back to your the demo that we just saw, and if it wasn't obvious, we cheated and did pre-record it. The demo that wasn't live, but this part is. But we did that yesterday, and I was trying to go to sleep last night and thinking about the whole day's activities of this recording and learning about machine learning, and it really occurred to me this pretty deep insight that I had about if I were in grad school again, or if I were an experimentalist, which I'm not. I'm just an industry cadfly now. But if I was actually doing real work, I would really be changing the way that I designed my experiments and changing the way that I collected the data, because
it really, I guess to be honest, before we actually did this whole SynBioBeta Live, I didn't really know what machine learning was. I knew it was a very exciting technology. I knew a lot of companies were doing it. I knew that software's eating the world. I knew that data's eating the world. Actually, through going through your example, Laura, now I'm like, oh, that's what it is. It's multivariate analysis and it's regression on that. But it's because you need to have your data in this format. Then you can apply this amazing new tool to it. And I've actually studied, I took courses in my master's thesis in machine learning 15 years ago, but never really put it in a context and never really had the opportunity to do that. What I think is important, and I think we missed this in the demo yesterday, and it really came out when I was watching it again just now, is this hidden variable aspect that you uncovered. You said that in the experiment that we saw there, we saw the things that they did monitor, which was pH, and they monitored strain, and that's what they were plotting on the old style data. But what really the machine learning uncovered was that because they'd collected temperature information and temperature data at the same time that they were looking at those fermentation runs, that's what came out, this hidden discovery from the data, that there's no way they would have found that unless they were looking at every single combination of variables.
Yeah, that's exactly right. I mean, gosh, how I would do things differently to go back now. I think I've learned so much after coming to Riffin about just how inefficiently I was collecting data before. And I was only making discoveries about things that I was intentionally varying because I wasn't looking at the data in the right way. And how much stuff was I missing? I mean, and how much stuff were buried in spreadsheets that are uninterpretable? I mean, we actually, when people would leave the company, we would literally reproduce their experiments because we couldn't understand the data that they had collected. And then once we did the analysis, we would do exactly what I showed you. We would plot it, and then we go, oh yeah, it must be strain. Like it must be, you know, all of the effect is coming from this one thing that I varied. But guess what? Most of the time, it's not true. And so, you know, you're just iterating on these really inefficient processes, and you're really slowing down the pace of innovation. If you can collect your data in a structured format, which by the way you can, it's pretty easy, then you can pull it all together, and then you can find out what's actually causing the variation, not just attributing it to this thing that you intentionally varied. So I would do things totally differently if I was going back to the lab.
A good friend of mine, Dan Rasky, who's at NASA Ames, they've got a new initiative in machine learning going on, and we were chatting about machine learning and chatting about what we're doing here. And he came up with a really nice analogy. He said that machine learning is like a new telescope that NASA's got, and you point it in a direction, and maybe it's a radio telescope, or maybe it's it's looking at some new wavelength of light. And you've got this new equipment, and suddenly you point it at data, maybe even old data that you collected in the past, and suddenly you can see all this new, all these new things in your data that you didn't see before. And I think that's exactly like machine learning. It is this tool. Are there any examples of people maybe going back through old data sets and then throwing them into Riffin and creating new insight?
Yeah, I mean, we do this for our customers as a proof of concept. We say, give us your historical data, and we will find trends in it that you didn't know were there.
Awesome. And
we've been able to do it every single time.
Got it.
Because that's how much people are really missing in their data sets.
Got it. Okay, great. Well, again, if you have any questions for Laura or for Tim, then please tweet at us or send them over email, and we'll ask them. And we're going to be looking in a short while at Epibiome and Aaron Hammock and how he's seen some of these new insights in the data. But before we do that, I've got a few other questions. In particular, can you tell me a bit about the speed of implementation? How long does it get people to be up and running using Riffin?
Yeah, so we can get people up and running in just a couple of weeks, maybe even faster. I mean, you've been involved with more implementations, Tim. Maybe you can give us an average here.
Well, if Laura does it, it's hours. Actually, quite literally, we've tracked our implementation time, actual hours spent. And you don't always work all these hours all at once, so it can spread over a few weeks. But the actual hours spent to implement Riffin and get a process and experiments up and running is as quick as 6 hours.
Okay, excellent. And then the whole 30 seconds, which is the title of this event, is because once you've got it set up, literally as you showed us in the demo, all this data flows from one process to another. So once you've got it all set up, it's automatic.
Well, two things. You can keep repeating that because it's a template, and you can keep executing on that template. But it, very, very importantly, the whole point of R&D is to change and improve and innovate, right? So that template cannot be static. So Riffin is designed with a versioning system, a version control system. You can change not only that template from day to day, hour to hour, minute to minute, you can actually adjust that template on experiments in process, so that if you forgot a variable or you need to make a tweak on it, you can do that too. You're not restricted like many of the data systems, legacy data systems are in making changes.
Can you talk a little bit about how you're seeing biologists turn into data scientists? Because in the past, I'm actually a bit of both. A software engineer, a really bad software engineer turned a really bad biologist, but having to kludge together these Python scripts that would collect the data. So, I mean, now you're seeing a lot of people, a lot of students coming through the education system, and they have got both. They can do Python and they can do wetware. They can do genetic engineering. But how are you seeing them, take these tools and then apply them?
So I'm gonna let Laura answer this, but I'm going to preface it because Laura came to Riffin having never done data science, never done programming. She was a chemist and a pharmaceutical scientist working for a pharmaceutical company, and joined Riffin. And now I would qualify her as a quite astute data scientist. And that happened in the course of a year. And she even wrote a blog post on Riffin's website that talks about that experience. I think it was called "Every Scientist is a Data Scientist," or
I think it's like "Be Your Own Data Scientist."
Be your own data
easier
scientist.
Than you think.
It's easier than you think. So I'll let
living proof that it's easier than you think. I think one of the barriers to me previously from becoming my own data scientist was that I didn't know how to code, and I still don't know how to code. So I'll throw that out there. But it turns out that you don't actually need to know how to code to be your own data scientist. So you did before, because you really needed to be able to code to pull together all of these data sets. But that's the part that Riffin is going to do for you. So we have this nice graphical user interface that allows you to just come in and enter your data, and then Riffin's doing all the hard part of joining it. Okay, so that's taken care of by Riffin. And then the next thing you need to be able to do is to script your analysis. And so for that, you do need to know a little bit of statistics, but it turns out that you can learn the basics in just a few hours, and then all it is applying that, and we can teach you how to do that. And then there's also programs out there like Jump that allow you to script your analyses without actually having to code. So you can just, you go into their user interface, you set up your analysis, and then you just say, save this as a script, and then you can run that over and over. And so I happily teach people how to do this. That's part of my job, and I'm really excited about teaching people how to be their own data scientists. I hate the siloed approach of I collect the data and then I throw it over the fence to the data scientist, because I'm the one that really knows the most about my data. I should be the one analyzing it, and I think that's really the power that Riffin is giving back to the scientists to say, don't throw it over the fence, just do it yourself. It's not that hard.
Yeah, I remember in my PhD spending, I think, about 9 months writing a whole Python script to connect everything I was doing together. And if I'd have actually just stopped and done it in real time, I'd have realized, that was the most important thing, to get feedback on the data immediately.
Yeah, exactly.
Not to write this automated script.
Totally. Yeah, and that's what Riffin is designed to do. It's to allow you to quickly iterate. So you put the data in, you can join it with all the other data that's been collected on that process. You can run your analysis and immediately see the effect of that, the effect of the whatever parameters were varied, and then you can just iterate on that and just keep improving your process continuously. You don't have to wait till the end. You should continuously be improving.
Got it. So if there's somebody who's watching and they're in a corporate R&D environment, what's the best way for them to kind of understand whether this is going to have an impact on them? And maybe tell me who in the organization do you think is going to be the advocate for this? And also, are there people in the organization who are going to resist this kind of tool?
Of course.
Yes.
Yes, I mean, change is hard, right? Riffin is introducing a change, and it's introducing a change on purpose. Because if you just keep doing things exactly the same way you always did them, how would you ever expect to see anything different come out of it? So change is hard. Some people find that transition difficult. Other people just
absolutely run and embrace it in seconds because they're so excited and they've been waiting so long to have something to do that. What we find is that it helps a lot in organizations to have some air cover from somebody at a principal investigator or a director level whose job it is to bring all these pieces together, and they create the organizational sponsorship and encouragement to take the time to go through that transition. If you have to do everything the same way you did it before and adopt a new software platform and a new process all at the same time, and nobody carves out any breathing room or space to learn some new tools, it's really hard to make that transition. So the air cover gives you space, and then the folks who are doing the science, an open mind and excitement about learning something new and trying something different is really what makes things tick. And it doesn't take long. You start, as Laura said, we see unexpected outcomes in the very first data sets that we collect, and we can start to clean up and improve your data in weeks.
Got it. Great. So hello@riffin.com is the email address that people should email if they want to set up a demo account, if they want to roll up their sleeves and try this out. One other thing that I wanted to mention was something that we're doing together in October with Amazon Web Services, and this is a satellite event where people could actually come. And can you tell us a bit more about that? Should people bring their laptops to that?
No, this is it's going to be a casual but stimulating conversation. We plan to have, it's about 3 hours during happy hour. Plan to have some appetizers, hors d'oeuvres, and cocktails. But we also intend to have some thought leaders give a few remarks, a panel discussion, and just hanging out and socializing. The plan is to have it at the Amazon or the AWS Loft. I'm not exactly sure the location, I forget. It's downtown San Francisco.
Great. Yeah, I think that's the AWS Loft in Soma, which I think isn't open yet, but it's going to be open by October. And this is with Elliot Menschik, who specializes in bioscience startups with AWS. But I believe also, and I'm just remembering this, I think we've got a workshop at SynBioBeta in October. So what I'm looking for is opportunities where people, if they're intimidated by what they see, how they can roll up their sleeves and get some help.
Exactly. We hope Laura or perhaps someone else from Riffin will be giving folks a taste of Riffin and a sense of what they can do with it in a short, in an abbreviated tutorial. It'll be open to anyone that joins or attends the SynBioBeta conference, and it'll be a great way to learn more, dip your toe in the water, so to speak.
Great. So I believe we have another question from the audience, and in a second we're also going to go to Aaron Hammock's the visit that we did to EpiBiome, because that's a really good example where you can see him actually dragging and dropping data into Riffin and seeing, even though there's a lot going on, and it's pretty easy just to take a data file and drag it into Riffin. So we're going to see that in a second. But yeah, so we have a question from the audience.
Yeah, one of the questions that's coming up is, does Riffin do DNA or molecular design?
Okay, so that's a good question. So yeah, there's a lot of BioCAD tools out there for dragging and dropping DNA design. Is that something that's capable of doing on Riffin?
So Riffin is able to track DNA sequences across your assays and annotate the data that you collect on whether this DNA sequence has an effect or not. It does not do molecular design, and that was a conscious, purposeful decision because there are software companies that specialize that and done a really fantastic job with such software, one of which is the Autodesk Genetic Constructor. And so instead of rebuilding and reinventing what already exists, we decided we'd rather integrate. So we are actually now with a customer integrating the Autodesk Genetic Constructor with Riffin to allow you seamless digital pathway from molecular design all the way to assay data. And we take the same principle, frankly, with analytics. Rather than reinventing Spotfire and Tableau and Jump and R and all these great analytical tools, we integrate and make it seamless.
And in terms of the speed of implementing Riffin, or the speed of doing this, how much faster is it than a legacy system for let's say data collection or analysis?
From my own experience, it would take 6 to 12 months to build a bespoke, custom database data system for a new process. So we had many instances where people would come up with a new assay, say I need a data system for this.
the IT group and the software group at the company was completely overloaded. And like, great idea. We can probably start that in 6 to 9 months, and it'll take us 3 to 6 to finish it and debug it and everything. With Riffin, as I said before, you literally can put a data system together like that in, for an expert, Laura is probably one of our greatest experts, probably 30 minutes to 60 minutes. Typically, it's about 6 hours.
Great. Excellent. So if you have other questions, email us live@synbiobeta.com or reach out on Twitter. So let's go over to Epibiome now and see how they are using Riffin. So I'm here at Epibiome in the Bay Area and with Erin Hammock, one of the co-founders and the CEO. Erin, tell us what's going on.
So here at Epibiome, we go hunting for bacteriophage in the wastewater treatment plants at the local Bay Area facilities. So this sludge is Bay Area wastewater. We filter it and then screen against panels of pathogenic bacteria, and the end result of our discovery efforts are vials of therapeutic compounds. So this is a vial of bacteriophage that is capable of killing ETEC, and ETEC is a huge threat to human health. So what's beautiful about the Riffin system, it lets us catch all of that in a seamless versioned manner that really gives us insight into what we've actually discovered. One of the excellent things about Riffin is this seamless capability to upload your data, specify exactly what happened in all of your data, and save it onto the cloud. So this is a large batch of experiments. It would take forever to manually enter it, and you associate it with a specific step in your process. For us, this is host range screening data, which fits into our larger phage discovery pipeline. And for the phage discovery pipeline, a really nice feature is you can actually specify with photos and detailed documentation exactly what needs to happen, which means that a new technician in the lab has a very clear track towards producing reproducible, replicable data. And that's excellent.
So as a process engineer, tell me why this visual representation makes sense for data collection in the lab.
What's great is having a visual system for capturing your process flow. You can really clearly see which steps are active, and then you can dive into them. And they're nested, and it all fits together in a really easy fashion to see what steps are part of which other steps. That's a huge advantage over a notebook.
So on the right here we have, this is an electronic lab notebook, is that right?
It's a process flow notes.
Okay.
So it's more what you would capture from your original designs.
Okay.
This is the step-by-step, what you want to do every day. And then where it does work as a lab notebook is it's versioned.
Got it.
So if your workflow, you change one of these, it logs the change, but you can always go back.
Got it.
And that's an advantage over pen and paper. You're not hunting through, your coworker who keeps bad notes notebook to figure out what they did last week.
Got it, okay. So if a new person's coming into the lab, how quickly do they get up to speed on this environment?
We've found that we can easily get people up to speed within a week or two.
Excellent.
It's a very rapid train time.
Great. Excellent. Well, Erin, thank you very much for having us today.
No, pleasure.
Great, so we have a few more questions coming in. Sorry for the echo there. We have a few more questions coming in from the audience, but before we jump to that, Laura, there's one thing that didn't really come up, but I know is important, is that there's no need to run separate tests to see if assays are working right. Can you explain a little bit more about what that means?
Yeah, that's right. So the thought is that if you are collecting your data in a structured format that Riffin allows, so you would just simply record all of your data in one place in the structured format, so that each of your experiments are just being collated together, so that every time you add an experiment, you can analyze it alongside all of the other experiments that you have run on that process. And so there'll be enough natural variation in the parameters as you just run your experiments during your normal day-to-day operations that you can start pulling out trends in the data. So there's no need to actually run specific experiments to optimize it if all of your data are in one place and you can just look at it in aggregate.
Got it. So it's kind of best practices that once you
Yeah,
set up your
that's
experiments,
right.
once you set up how you're going to collect the data, what you're going to collect about it, then you can be you can be collecting and analyzing it in real time, even if you're not doing the whole pipeline yet.
Yeah, that's right. And instead of sticking it into all these individual Excel spreadsheets, if you put it into Riffin, it'll just automatically be joined to all of the other data that were collected on that process. And so, yeah, like you're saying, you can just continuously iterate.
Got it. And we saw from Aaron Hammock just now at EpiBiome how he did just drag a file, throw it into Riffin, and away you go.
Yep.
And what what Aaron really underlined when we had a discussion after we visited was the hidden discoveries that he found. So maybe you can talk a little bit about that in terms of false positive discovery and why that's impactful.
Yeah, so I mean, I think we have talked about this a little bit, like how you can only discover things if you're actually collecting the data on it, right? And if you're putting it into your analysis, and so yeah, I mean that's really what we enable. Like you saw in the demo, how we had a false hit there, so it looked like strain 4 was the best, but it was only because it was confounded with other variables that we weren't accounting for. And so when you're able to parse that out, then you're able to find the actual result among the noise.
Got it. Okay. So I think we have a few more questions from the online audience. Kevin, who have we got online? What are their questions?
Yeah, so Vaishali Theor, I'm sorry if I'm mispronouncing the name, from Imperial College asks, the Riffin process involves the development of a robust optimization model. And he's very interested in how the computation is so fast, that getting to that 30 seconds. I wonder if you can unpack that a little for us?
Sure, I'm happy to. So I think it's important to note what Riffin does. Riffin is designed to design your experiments, collect the data, assemble the data, join and connect and link data points across the sample flow, and then present that resulting data in a fully articulated, fully annotated, structured data table ready for statistical analysis. That's Riffin's job because that's where machine learning and data scientists folks typically spend 80% of their time. In fact, we've had some customers say it takes them 2 months to pull together the data for one experiment. Riffin can take that literally from data collection to data ready for analysis in less than 30 seconds. Riffin doesn't do the machine learning itself because you can get that from Apache Spark or Amazon Web Services or R or JUMP or SAS or a million different places. So the actual computation of the model is really up to the algorithms that you choose to apply, although that's very fast. Believe it or not, when Laura did her demo, she was doing the regression analysis in real time. It was actually computing that, and it took less than a second. So for simple models or relatively small data sets, it's a split second. If you had 1 billion data points, it might take longer than 30 seconds to run that model. But that's usually not how much data people have in a laboratory setting.
Great. Let's take the next question.
Yeah, I've got a lot of questions from YouTube right now. What is the purchasing model? This is from Maria at University of Edinburgh. Is this a one-time purchase or a subscription model?
It's a subscription-based model. It's all-inclusive. If you buy a subscription to Riffin, you get an account. You're up and running as soon as you can open your web browser. There's no installation. We do continuous improvements, upgrades, maintenance, software, server, hardware costs. It's all included. Help desk, support. You can call us up. You can email us. That's part of the subscription, and it's why we do that way. And we release upgrades and improvements roughly every 6 weeks, so the software just keeps getting more and more powerful, and you don't have to worry about paying buying the software again. It's just part of the subscription.
Great.
Excellent.
Another question coming in is from Carlos at Biosynthia, and he asks, in some particular R&D pipeline, such as cancer research, both public and private, there's a crisis of reproducibility of experiments. And it seems to Carlos that Riffin could solve this problem. Do you have any ideas for how you could address that issue potentially?
Do you want to take that, or?
Yeah, you take it.
Okay. It was honestly a lot of why Riffin exists. It was to address that problem. But I'll be frank, reproducibility sounds like a kind of a boring thing. Discovery is pretty cool. And reproducibility isn't really about repeating your experiments over and over again. It's about quality assurance. Well, let me give you an analogy. The manufacturing industry in the 50s used to produce two cars from their pipelines and throw one away because only one of them actually worked. And that was a quality control, reproducibility mentality. Repeat it if it doesn't work. Then the Japanese introduced total quality, and they said, no, if you're waiting till the end, it's too late. You need to go upstream into the manufacturing pipeline and fix the problems before they propagate. And then 90% or 99.9% of your cars work perfectly when they come off the production line. And your productivity as an organization leaps. It just doubles, triples, quadruples. They actually got a quadrupling or a tripling of productivity in manufacturing. That's the approach Riffin takes. If you're waiting till the end of your experiment to think about reproducibility and quality, it's too late. You have to fix the process by which you generate the data before you generate it. And that's what Riffin's designed to do.
Yeah, and I'll just add one more thought, that reproducibility can really come from two different sources. So the first is that I can't reproduce what somebody did because I literally do not understand what they did. And so when I'm trying to reproduce it in my lab, they did not record enough information for me to reproduce this. And the second is that you attributed the result to you, it was incorrect. The variable that you said was causing this effect was not actually the variable that was causing that effect. In the example I gave, you said, yep, strain 4, that one's definitely going to give you the highest succinate yield, but guess what, it doesn't. And so that is another source of your reproducibility. And Riffin actually solves both of those by very clearly visually laying out exactly how an experiment should be performed, and then by also giving you the tools by which you can figure out what are the underlying causes of the outcome that I'm seeing.
Great, excellent. Any other questions online, Kevin?
Yeah, one kind of thematic question that is coming up is, how exactly does Riffin make data entry easier? How do you ease the data into the system?
Yeah, so we think a lot about this. So we realize that the software is only useful if the scientists are willing to use it, right? And so our goal is to make it as easy as possible to get data into Riffin and to eliminate any transcription, any copying and pasting. So one of the ways in which we do that is that we allow you to register entities in the system and then associate metadata with that. So let's say that I'm using a particular cell bank in my experiment. I can just assign that cell bank from my inventory, and there'll be lots of metadata that just comes into the experiment automatically. So something like the lot number or the strain name or the organism. That's the metadata about that sample that just will come in. I don't have to do any copying pasting. And then as we saw Aaron demonstrate, you can see that you can just very quickly take an instrument file, upload it to Riffin, and then all the data just comes in. Again, no copying and pasting. So we really work hard to make this tool work for the scientists.
Excellent. Yeah, probably the final question online.
Yeah. Sure. So the last one might be, is this the end of pivot tables?
Well, so when you say pivot table, I think of Excel. And frankly, I and many people have a love-hate relationship with Excel. It's a great tool. It does amazing things, but it also gets overused for the wrong purposes. So a pivot table, there's nothing wrong with it. We use them. And for certain questions, it helps. But the problem is it gets overused as the only way to answer questions. And so I don't think, in my experiences in the past 10 years for scientific analysis, I've used a pivot table once. I usually go to a statistical tool. I look at distributions of variables. I look at variance decomposition, regression analysis, correlations, etc. And I do it in a visual way. I have to see the statistics, but I have to see the plots and the correlation plots. It's amazingly powerful to how your brain can not only see the relationships when coupled with the statistics, but it also helps you detect outliers and deviations when you look at it visually. So one of the cool things we have done with Riffin
is through the API that's accessible to anybody, not just ourselves, but anybody who uses Riffin, we've actually created bidirectional flow. So you can pull the data through the API into a program like JMP. You can visually analyze it with statistics. You can select outliers, tag them, push the data back into Riffin so that your original data set is now annotated with what you saw was an outlier or not, good or bad data points. And that's a far cry from a static pivot table in Excel, which I don't want to disparage Excel, but it's limited in what you can do.
Great. Yeah, we have
one more quick question. Is Riffin GDPR compliant? It's a hot topic.
Riffin is both compliant with GDPR, but it's also 21 CFR Part 11 compliant, which is the electronic data systems requirement for the FDA and a similar one for the EMEA. The one thing I want to say about GDPR, Riffin doesn't collect any personal data. Very, very intentionally, our business model is not about using or harnessing or maliciously investigating your data. We don't touch it because this is your R&D data. So we don't collect any personal information other than a login, a username, and a password. So it makes GDPR really easy for us.
And so it brings up a good point. A lot of biopharma companies are going to be scared in terms of the security of their data, putting it into the cloud. I mean, has that paradigm shifted? Has the industry changed?
I would say people are sprinting to the cloud.
Great, excellent.
It's more secure, more stable, more robust, more scalable. When the size of the data grows, you can horizontally scale, add servers and parallel compute power. From a security standpoint, Riffin wrote its first security document before it wrote its first line of code. And we actually achieved the highest level security approval for one of our customers. So they have multiple tiers. That's super top secret level. We were approved for that because we take security so seriously.
Excellent. Great. Well, this has been a fantastic first show. I wanted to thank Tim for both being Tim and Laura for being the guinea pigs on the show. I don't think we achieved Wayne's World status.
Next time, guitar and drumstick.
Yeah, exactly, but the live studio audience has helped. So if you want a demo of Riffin, hello@riffin.com is the place to email. We would also love your feedback on SynBioBeta Live. This is the first one that we've done. We have another 2 products coming up. We're doing a deep dive with the lab site Echo on Wednesday, June 27th at 8 a.m. So we'll be with Chance Elliott, who's the head of automation at Amyris, and he's going to be talking through how he's using the lab site Echo to reduce costs in terms of their next generation sequencing prep in their DNA assembly prep, so I hope you can tune in for that one. We're also going to be live in New York with Opentrons, and I'm very excited to talk about the launch of their OT2, their new liquid handler. That's going to be in July. Also, I wanted to plug Tim for a couple of reasons, because one, you were one of the trailblazers that we interviewed for our book, What's Your Bias Strategy? So if you want to go a bit deeper on Tim's philosophy and history and passion for the field of synthetic biology, then I encourage you to read the interview in the book. And then also, you're going to be keynoting in October at SynBioBeta. We got a couple of ideas. We haven't quite finalized it. One was 20 years of synthetic biology. But what was the other topic? Have you had time to think about what you're going to be talking about?
not had time to think.
I'm springing it on you, so no worries.
I think we're on the verge of a digital transformation, or actually in the midst of a digital transformation in biology, and inevitably I will speak and add some color to some of the comments that we discussed today.
Great. I really think we are. We really are on the edge of something big here. Great, and so I would also love dearly your feedback on SynBioBeta Live. You can email me directly, john.cumbers@synbiobeta.com, or on Twitter, John Cumbers. And I'd love to know what you liked about the show, what you didn't like about the show, what we can do better, and what you'd like to see more of, because this is something that we think, this kind of niche network for you, the thought leaders in the synthetic biology industry, the people who are out there in the field, making biology easier to engineer. We want to do more of these kind of activities. So it leaves me to thank Aaron Hammock, who's the CEO of EpiBiome, who you heard from, John Conway of Accenture, for some wonderful insights into how larger corporations are looking at their R&D organizations. Thanks to Garrett Kushner on the SynBioBeta team and Kevin Costa and the rest of the team for really making this production. You can't see what's going on behind the cameras like we can, but there's quite a production effort here. And finally to Tim and Laura for making all this happen. So
Thanks.
yeah, really appreciate you guys coming in, and hope you've enjoyed it.
Yeah, definitely.
It's been awesome.
Great. We'll see you again next time. Thanks for joining us.